what happens in pro, meta, ana and telo. See, in prophase, the chromosomes have become short and thick and are clearly visible inside the nucleus. Okay, now each chromosome has already duplicated to form the chromatids. Now I am following page number 13 of the prophase part in the second column. You open your book and please follow what I am trying to teach you. What happened in prophase? The chromosomes have become short and thick and clearly visible inside the nucleus. Each chromosome has already duplicated, having made its copy to form chromatids. Two sister chromatids remain attached to each other at small region called centromere. Now I have already discussed centromere in my previous one. The centrosomes splits into two along with simultaneous duplication of the centrioles contained in it. The daughter centrioles moves apart and occupy opposite poles of each cell. Now one thing is that what is pole of each cell? What is pole of each cell? So you need to understand that. See, for that please move to page number 14. Okay, here you will find that both plant and animal cells are given beautifully, and it has been described that how these parts are separating and what is happening. So suppose this is a cell. Okay, now this is a nucleus. What we call them as the pole. This part, these two parts are called the pole. See, on the starting stage, or when it is in interphase, or it is in resting phase, what happens? So the chromatids are like this. Okay, it is like this. Now when cell division is occurring, so what do we understand by the term division? It means that it will get divided, right? So what happens? All the things that we are learning here is given here termed as place, things and everything. What happens is that these two are the equators. So slowly these chromatids gets to from this part and it gets separated to here. Okay, it moves towards the equator, and as it moves towards the equator, replicating of the parts, the duplicate ones, this all gets formed over here, and when it goes on like this, after a certain time, when the resting phase or when all the phases are completed, this nucleus duplicates. Okay, what happens? It gets stretched away. So what it will look like, students, if we like stretch this part like this? So it will look like, see, cell is only one part. Okay, now this gets elongated like this, and this is the equator. Okay, these get stretched. These are the lines I am marking. So these get stretched, and what happens after a certain period? We will study about this. After a certain period, in a single cell, two nucleus get formed, and when two nucleus get formed, this particular thing is known as karyokinesis. That is division of nucleus. Am I clear? And what is cytokinesis? Cytokinesis is when these cytoplasmic material also get divided. So what will happen? See, this, suppose this is a cell, two nucleus have already formed here, and after that the cytoplasm will also get divided and two new cell will get formed. Okay, this is the basic concept of cell division. This is what cell division is. Okay, now this is under mitosis, this is karyokinesis. Karyokinesis is the division of the particular nucleus and nuclear material, and cytokinesis is the division of the cytoplasm including all the cell organelle. Okay, am I clear up to this? So you go through the diagrams that is given in page number 14 of ICSC Concise Biology class 10, figure 2.1, that is the cell division phase. You will understood it clearly. If there is any confusions, I am here to make you understand how the thing is happening. Yet I am making you understand each and every part of this prophase, metaphase, anaphase and telophase. Okay, so what happens in prophase, that I have already told you. The nuclear membrane and the nucleus disappears. The duplicated chromosome starts moving towards the equator. So this is the equator. This is the equator. Okay, it starts moving from here to this particular place. Okay, now coming to metaphase. The duplicated chromosomes arrange on the equatorial plane. Each chromosome gets attached to the spindle fiber by its centromere. This is metaphase. Now coming to anaphase. What happens in anaphase? The centromere starts attaching the two chromatids, divides, and the two sister chromatids of each centromere separates and are drawn apart towards the opposite poles. The drawing action is performed by contraction of spindle fibers. Now, what are spindle fibers? Spindle fibers are these. Wait, let me show you what are spindle fibers. I am rubbing this one. This is looking very funny. Anyway, you need to understand. It doesn't matter it looks funny or not, but what you need to do is you need to understand. Suppose this is the cell. Okay, now this is the nucleus. These are what? These are chromosomes. So how many humans have? They have 23 pairs. Okay, they are in pairs. Anyway, we are taking an example. What happens? On the first stage, it is like this. It is ready for cell division. See, chromatin fibers now have become chromosomes. They are thick and we can clearly visible them. They are clearly visible and you can clearly see them, understand them. Now they starts moving towards the equator. Slowly they starts moving towards the equator. They starts moving towards the equator. Now spindle fibers appears between the daughter centriole, forming chromatid spindle. The nuclear membrane and the nucleus disappears. These are in prophase A and B. So what happened? Aster formation take place. What is Aster formation? Can anyone tell me what is Aster formation? See, Aster formation is this formation. This is known as Aster formation. What happens? I am again repeating prophase A and B. You will just go through your book, page number 14. Centriole starts moving apart and reach opposite poles. Okay, it reaches the opposite poles. Chromosomes become distinct, duplicated as paired chromatids. Sister chromatids attach to each other at a small region called centromere. I already told you. Now, spindle fibers appears between daughter centrioles, forming a chromatid spindle. New membrane and nucleus disappears. Okay, so this is what happened during prophase A and B. After that we go to metaphase. Each chromosomes get attached to spindle by its centromere. Chromosome lined up in one plane at the equator. Here what happens? Here this lined up here in a mid plane. This get lined up. Okay. Now anaphase, centromere attaching to two chromatid divide splits. The two sister chromatids of each chromosome separates and are drawn apart towards opposite pole, pulled by shortening of the spindle fibers. A furrow start in the cell membrane and divide in the middle an animal cell. So what happens when this gets elongated, as I've told in the previous diagram, this get elongated. In the mid, a furrow generates. Okay, I will showing you again. So this round shape goes to this like shape, and here what happens, a furrow appears. It's just like an apple. Okay, all of you have eaten an apple, I guess. So this furrow appears, and this furrow, what happens, then again it becomes like this. Okay, and from here the division take place. So in telophase, that is the last phase, what happened, each chromosomes or each chromatid or the daughter chromosomes length then becomes thinner and turns into a network of chromatin threads. Then again it restores to its previous shape that it was there. Nuclear membrane reappears, that which have already disappeared, it again get reappeared, and nucleus reappears in each daughter nucleus. So this is how cell division occurs. Now see, wait, it is not yet the cell division, it is the nucleus division, the karyokinesis. Now we will go to cytokinesis. At the end of the telophase, a furrow appears in the cell membrane in the middle, which deepens and finally splits the cytoplasm into two and thus producing new cell. Karyokinesis and cytokinesis, please go through the box provided in your book. All the nuclear changes that occur during cell divisions are collectively termed as karyokinesis. Karyokinesis is followed by the division of cytoplasm, which is known as cytokinesis. So students, am I clear up to this? I guess this is all about the mitosis inside cell division of your chapter 2, cell structure of chromosomes and cell division. Okay, so this is all about mitosis. I am strictly following Concise biology class 10. You are all requested to go through that particular book. It is given here in a very detailed manner. You all can understand if you go through this video properly and you follow your book. This chapter, I hope and I believe that particular chapter will be very much clear to you all and you will have no doubt at all. So moving on, differences between mitosis in animal and plant cell. This is a common difference that we all will get. Quickly look into table 2.1 of page number 15. You will find that mitosis in animal cell, asters are formed. Mitosis in plant cell, asters are not formed. In plant cell, asters are not formed. Please keep this in mind. Okay, cytokinesis by furrowing of cytoplasm. The furrow starts from the cell membrane, which then proceeds to the center of the cell. And here, cytokinesis is by cell plate formation. So we all know that plants have cell wall and animal cell do not have cell wall. We all know that. So here what happened, here furrowing of the cytoplasm take place in animal cell, but in during plant cell division what happens, cell plate formation take place. Okay. The third point, occurs in most tissue throughout the body for growth and replacement, and in plant cell what happens, occurs mainly at the growing tip for the lengthening and side for increasing growth. Okay, now one more thing that I have already questioned you, I don't know you all remembered or not. The thing is that where does this mitosis take place in our body? So where does mitosis take place in our body? Mitosis take place in our body during all the repair and replacement of the cells that continuously lose or get died from our body. How many of you know that if we just scratch our skin, we'll get thousands and thousands of dead cells in our nails? Do you all know that? Yes, we will get that. Okay, so now these cells which are continuously dying, or suppose in case of any of the injury that we have in our body, what happens, or during any cut or any wound, the cells need to get replaced or repaired, and it is done with the help of mitosis and not meiosis. Meiosis is something different. We will go through it in our next video lecture. Okay, so This is all. Any other point? Let me quickly see. Okay, significance of mitosis is given here, page number 16, 2.7.3. It is for growth, repair, replacement, asexual reproduction, in which the unicellular organisms such as amoeba or the yeast cell divide into two. Okay, maintains same chromosome number in daughter cells. It maintains the same chromosome number in the daughter cell as well. So this is all about mitosis. I hope you all have understood that. In the next video lecture we'll continue about meiosis. See, the main reason why I am not continuing my lecture all total very long video of 1 hour or 1.5 hours is all because I can continue with that, but you all cannot understand that. Now if I tell you that we have everything to it, from starting from say morning till night, and I give you everything at once. Okay, I give you breakfast, I give you lunch, I give you supper, I give you dinner, everything at once, you will not be able to eat that, right? So that is the reason I am giving it part by part, so that you all can understand this particular chapter, as this is very important for your upcoming ICSE examination. So as per now of chapter 2, structure of chromosomes and cell division, we have already studied about what are chromosomes, what are the structure of chromosomes, how we can define them, what are chromatids, what are chromatins, and what does it consist of. It consists of DNA and histone proteins. We have already studied about that in our first video lecture. In the second video lecture you have understood about mitosis, and I hope that you all have no queries regarding mitosis. So coming on, we now have meiosis. See, in ICSE syllabus meiosis is very less, and we just need to study that, but I will let you understand how meiosis happens up till it is in your syllabus. Okay, anyway, so what is meiosis? Meiosis is the reduction division producing gametes. So meiosis is reduction division. Okay, reduction division. Why it is known as reduction division? See, let me go through the book one. Please open your book, page number 16 of Concise Biology part 2, that is Concise Biology class 10. Okay, of Selina publisher. So we will find here meiosis. Meiosis is a kind of cell division that produces the sex cell or the gametes. It take place in the reproductive organs, that is testis and ovary, in human to produce sperm and ova. In the flowering plants, it takes place in the anther and the ovule to produce pollen grain and female gametophyte. The most significant aspect of meiosis is that the number of chromosomes in the sex cell is halved. For example, out of 23 pairs of chromosomes in humans, only single chromosome, that is one member from each pair or haploid cell, take place. Okay, what happens? So let's see. Suppose this is a cell and this is a nucleus. Here we have chromosomes. Now these chromosomes are in pairs. Okay students, these chromosomes are in pairs. Now what happens? This is, suppose we are telling it as 2n. Okay, whatever be the number, be it 2n. So what happened? This gets divided into two parts. Okay, into two parts, where this 2n, what happens? Suppose this 2n, what happens? Here it is 1, 2, 3, 4, 5. See, here is 5, that is 10 number of chromosomes. Okay, we are taking it as an example. What happens? From here it is, we can say it is 1, 2, 3, 4, 5, that is 5 pairs. Now each chromosomes from these 5 pairs comes here and comes here. Okay, so this will form 1, particular n, and this will form another particular n. So this 2n gets break to n and another one n. Okay, now this n gets fused, this n gets fused with another n that is coming from another parent. Same for this one as well. Okay, same for this one as well. Say suppose this is parent A and this is parent B. So here is also 2n, it is n, it is n. So what happens? This is a reduction division, because see, from here to n only n comes, single pair comes to this one and this cell. Now here also from parent A the same thing happened. Now these two cell, one is from parent A, another one is from parent B, these two get fused together, and these chromosomes get altered and they form a new cell which consists of the same number, here it is n, the same number, that would be n plus n, and it would be 2n. Thus the number of chromosomes get restored, and as this particular thing is happening we call them as reduction division. Okay, and this particular meiosis is occur in our during reproduction. Okay, so the most significant aspect of meiosis is that the number of chromosomes in the sex cell is halved. For example, out of 23 pairs of chromosome in humans, only a single chromosome, that is one member from each pair, are passed to the sex cell. This is essential because when the male and the female gametes fuse during fertilization, the normal double number of chromosomes in pair is reacquired. Definitely, as I have told you, that is n and n, this will become 2n. Okay, I hope this meiosis is clear. So we will continue with the significance of meiosis. What are the significance of meiosis? You can follow page number 17 of Concise Biology Part 2. Chromosome number is halved in gamete cell, so that on fertilization the normal number 2n is restored. It provides for mixing up of the genes, which occurs in two ways. Okay, what happens? The matrial and parental chromosomes get mixed up during first or the reduction division as they separate from the homologous part. What happens is that the matrial and the parental chromosomes get mixed up. Okay, so the thing or the parental chromosome, that it comes from both the parents, right, as I have already told you. So from both the parents it comes together and it gets mixed up. Second, while the maternal and the parental chromosomes are separating, the chromatids material very often get exchanged between the two number of homologous pair. This is known as crossing over, which result in genetic combination. Chiasma in the X-shaped structure formed due to the crossing over between the non-sister chromatids of the paired homologous chromosomes. Okay, now what happens is that, okay, let us study this. I hope you all are interested and you all are getting interest in this particular chapter. So suppose this is one chromosome, this is another, say another chromosome. Okay, now what happens? Suppose this part, okay, this leg, see this leg, this particular part, it gets crossed over to say this particular part. Okay, so this is known as crossing over. What happens in crossing over? This is nothing but the chiasma, or the plural what we pronounce it as chiasmata, is the X substructure formed due to crossing over between the non-sister chromatids of the paired homologous chromosomes. So these are chromosomes. When they get, like, see, n number comes from one parent, another n number comes from another parent. When they get fused together, sometimes what happened, some of the part of the chromosomes get crossed over to another pair or another part of the chromosomes. Okay, so chromatin exchange part, or these are the chromatid exchange part, what we will call. Okay, so quickly moving over, the mixing up of genes during mitotic division provides for the innumerable variables in the progeny. That is how the children of the same parents, however similar, are different from each other in certain respect. Okay, this is also a very important part. You will find here what it is telling is that mixing up of gene during mitotic division. Okay, in mitosis we have already studied. So this is only because of the mitotic division, while your uncle or your father, they both are from your single grandparent. Okay, but yet they differ from each other a lot. There may be resemblance of the same thing, but yet they differ from each other. This is due to these genes that coming during mitotic division. And in meiosis, what happened? This crossover only take place. So you will also find a difference given between meiosis and mitosis. So you will find that in page number 18, table number 2.2, difference between mitosis and meiosis. So I hope this ends up the chapter of chromosome, and I hope everything is clear to you all. Go through the questions. I will definitely provide you the notes. Go through the questions, and I believe that if you have already watched this video with your book keeping aside, everything is clear of this particular chapter, chapter 2, structure of cell and structure of chromosome and cell division. So thank you. We will continue, and I will provide you the notes and inside questions of this particular chapter. Thank you.